Namaste students, Khushro Vidya. I, Iam Dr Chandrakala, Assistant Professor, Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the embryo culture. Embryo culture, a seed plant embryo, is a part of a seed consisting of a precursor tissue for the leaves, stems, roots, as well as one or more cotyledons. The young sporophyte of a seed plants usually comprise into a rudimentary plants with plumule, radicals, and cotyledons, which you normally see in a normal germinating plant. So embryo culture, what is the requirement of an embryo culture here? Embryo culture similarly can mean that the growth of any species during its embryonic stage in an artificial medium in order to enhance its livelihood or uh, survival rate, increase its survival rate. We have to do an artificial medium, have to create an artificial medium for the embryo to grow. In certain conditions where the embryo is not able to grow due to certain specified reasons, so we try with embryo culture here to develop the plantlets into in an artificial medium. So the embryo culture can also mean the use of artificial medium for embryonic growth during the in vitro fertilization, which can last for 2 to 5 days, and thus which the embryo is transplanted into a surrogated mother as a animals or a human being it was done. Okay. In the same way, in plants also, the embryo rescue is being done in the in vitro culture techniques with where the development of a plant embryos that might not survive and become viable plants. So here the embryo rescue plays an important role in the modern plant breeding and ornamental plants or the crop plants, certain crop plants, where we have to rescue the embryo. So the most widely used rescue procedure is referred to as the embryo culture and involves the excising of the plant embryos and placing them into a specified medium where it can able to grow properly. So for embryo culture, embryos are excised from immature seeds and infusing into a medium, providing an aseptic and sterile area. The culture plates or the culture tubes with excised embryos are transferred to a culture room maintaining at a suitable temperature, photoperiod, humidity, frequency, and excised embryos that give rise to a seedling generally varies greatly here. So the advantage of the embryo culture is recovery of the distance of the hybrid, recovery of the haploid plants from the interspecific crosses, propagation of the orchid, or shortening of the breeding cycle, overcoming dormancy in addition to the ovules or the ovary can be cultured. So the importance of here embryo rescue and embryo culture is these techniques helps to develop the plant that are viable. The embryo rescue plays an important role in the modern plant breeding and which allows the development of many interspecified or intergenic food and ornamental plants of a crop hybrid. So where this technique nurtures immature or a weak embryo, thus allowing the chance of survival or increasing the number of chance of surviving. So the tissue culture technique in combination with the molecular technique has been successfully used to incorporate a specified trait through a gene transfer. So in vitro techniques for the culture protoplast, embryo culture has been created with new genetic variations in the breeding line. So the factors are like embryo are manually excised and placed in the immediately into the culture medium and they provide a proper nutrient for the survival and growth. It depends upon the time of the culture, composition of the media, or the two types, mainly like morass and schools medium, that is MS medium, and V5 medium, Bridging medium, where it is like the composition of the media will vary in terms of the concentration of the media, supplements requirement, and the generally depend upon the stage and development of the embryo. So the young embryo would require a complex medium with high sucrose concentration, while more mature embryos can usually develop into simple medium or a low level of sucrose. So the temperature and light, which varies in a species to species, depend upon the cool season crop or a low temperature for the warm season crops. So the embryo sometimes grows between best maintained in a dark or 1 to 2 week in transfer into the lighted for the chlorophyll formations. So the techniques and molding here, the embryos are located in a sterile environment for the ovules and the surface sterilization takes place. Now, embryos are where the embryos is not necessary much, or the embryos are removed aseptically from the environment, from the surrounding, or the embryo are often well protected by surrounding in tissues or harsh environment, or from surface disinfectant. Or direct disinfection of the embryos needed if the seed coat is cracked or endophytic pathogens exist here in the seed coat. So here the small embryos requires for the use of the micro dissecting tools and the dissecting microscopes for the exercise of the ground injuries. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur.